Okay, this first video of the first lecture in block 1 is going to be a little bit short. The reason being, normally when we do this, I've allowed you time to go through the syllabus and the calendar, which you've already gone through those 2 videos. So today we're going to do a little bit of a review of the mechanisms of intercellular communication. Intercellular implies that it's between 2 cells. This is table 18-1 on page 612 in your textbook. When you look, this table is set up that shows the mechanism of intercellular communication, how it's transmitted, what the chemical mediators are, and the distribution of effects. If you've had me before, you'll know that this is going to be a compare and contrast question. Whenever we say a compare and contrast question, we say how are they alike or how are they different? What's the best way to do a compare and contrast question? By a chart. This is set up in a chart form that is a comparison. So this would be a good thing to learn so that you would be able to do well on that essay question. So let's start with direct communication. Direct communication, you can see by that first diagram in the upper left-hand corner that the cells touch. So that when we talk about direct communication, adjacent cells, or those cells that are next to each other, have to be touching. They transmit that information through gap junctions. We specifically talked about a specialized gap junction in 175 that was between cardiac muscle cells. Can you remember what those gap junctions are called? If you said intercalated disc, give yourself a pat on the back. So through those gap junctions, we have chemical mediators that are going to be either ions, which you know mean electrolytes in biology, or we just talk about them as charged particles, small solutes, or lipid-soluble materials. So now you can see that when we talk about gap junctions, we're talking a lot in 176 about the 3 big ions: sodium, potassium, and calcium. You remember sodium and potassium are Nike. Sodium in, potassium exits. That's always how we generate an electrical impulse. Calcium, I'm sure you remember from 175, is necessary in all types of muscle contraction. So some of the ions are going to be sodium, potassium, and calcium. Well, how are those effects going to be distributed? Bottom line, it's adjacent cells. They have to be next to each other because they have to have gap junctions. So when we talk about direct communication, it's going to be through gap junctions. We are mostly going to talk about ions, classic examples, sodium, potassium, and calcium, and it's going to be a communication that's limited to adjacent cells. If you look at the next row, paracrine communication. If you remember, para in Latin always means around, and crine, C-R-I-N-E, means secretor. So when we talk about paracrine communication, it is an around secretion, or it's easier to think of as a local communication. When we talk about paracrine communication, we're going to have paracrine factors, there's a shocker, that are going to go through extracellular fluid. We talk about that extracellular fluid. Most of the time, that's going to be interstitial fluid, or ISF. And when we talk about paracrine factors, we talked about one in the first semester. The classic is prostaglandins. So that when we talk about the distribution of effects, they're going to be primary, and that was flash for those of you who remember, primarily located in a very small area, and you have to have a target cell. A target cell has to have a receptor. So that's another important thing to remember about paracrine communication. target cells, or those affected cells, have to have appropriate receptors. The third type of communication is called endocrine communication. Endo in Latin means inside, crine means secretor. Endocrine communication occurs through the bloodstream. What I want you to make sure that you remember is it just doesn't dump in the bloodstream. It always has to go through interstitial fluid, then it's going to go into the bloodstream. So remember, when it says bloodstream, you need to, in your head and on an exam, put that it goes first into interstitial fluid and then it's going to go into the bloodstream. Those chemical mediators are called hormones. And when we talk about hormones, there's several things that you need to remember. The first is that they are chemical messengers. That means that there's some kind of ion or small organic molecule that talk between the two cells. We are going to talk about this more as we delve into the endocrine system. You need hormones in small amounts. Always think of hormones as being very similar to enzymes. You need them in very small amounts because they're reusable. And then in endocrine communication, when we talk about the distribution of effects, the target cells are primarily going to be other tissues or organs, but they also have to have a receptor. So endocrine communication, inside secretors, they secrete those chemical messengers called hormones that are required in small amounts because they're reusable. They're going to go into interstitial fluid first, then through the bloodstream, and their target cells have to have appropriate receptors because they're not touching. Are you seeing a common theme here? If they don't touch, they have to have a specific receptor. And then the very last type of intercellular or between cell communication we did first semester, synaptic communication. Well, here's a shocker. Synaptic communication occurs across synapses. The chemical messengers are neurotransmitters. We talked extensively about two neurotransmitters last semester in 175, ACH or acetylcholine and MAO or monoamine oxidase. Those are the two types of neurotransmitters we talked about. Just briefly, remember acetylcholine goes from a motor neuron to a skeletal muscle, and then MAO, monoamine oxidases, go to smooth muscles. And again, synaptic communication is going to be limited to a very specific target cell, and they have to have a receptor. So how are you going to think about these four types of communication? First, you're going to memorize the name: direct, paracrine, endocrine, and synaptic communication. And then let's look at it logically. How are they transmitted? Direct communication through gap junctions. We think of intercalated discs. Paracrine communication through extracellular fluid, which essentially means interstitial fluid or ISF. Endocrine communication, it's going to go into interstitial fluid first, then it's going to go into blood. And then synaptic communications occurs across a synapse, which you know is that space between a neuron and some type of muscle, in this case. When we talk about chemical mediators, direct communication, think of ions or small solutes. We always think of the big three ions, sodium, potassium, and calcium. For paracrine communication, they call them paracrine factors. That's going to be prostaglandins. When we talk about endocrine communication, it's hormones. We're going to delve into those at great detail coming up here in the next lecture. But they're chemical messengers that you need in small amounts because they're reusable. And then we have synaptic communication across synapses that uses neurotransmitters like acetylcholine and monoamine oxidase. So the third thing that you're going to think about is the distribution of effects. You need to think about it in distance and why. So direct communication occurs only when cells are touching. So it's a very, very short distance, in essence, none. And the cells have to have junctions between them. The last three, paracrine, endocrine, and synaptic communication, all have to have receptors because they don't touch. those chemical mediators are going to be specific for the target cells. When we talk about paracrine communication, it's locally distributed. When we talk about endocrine communication, it's going to go over very long distance. You know that because it's talking about the bloodstream. And then we talk about synaptic communication, it goes over that short distance called a synapse. So now we're going to delve into the endocrine system and what its different organs are. This is Spotlight 18-12 on page 615, and also the figure in front of you is figure 18-1 on page 614. If you look at this picture, you can see that we have the endocrine organs, hypothalamus, pituitary, which has two parts, the anterior pituitary or adenohypophysis and the posterior pituitary or the neurohypophysis, the thyroid gland, the adrenal glands, the pancreatic islets, the pineal gland, and the parathyroid glands. Then we have what they call secondary endocrine organs, and those have some other specific function. We talk about secondary endocrine organs. We talk about the heart, the thymus, the adipose tissue, the digestive tract, the kidneys, and the gonads. The gonads involve the testes and the ovaries. So when we talk about the endocrine system, I want to go back to one thing before I forget. Many times people get the thyroid and the thymus confused. There's an easy way to remember it. The thyroid gland is like a bow tie for your larynx, and your thymus gland is like a western tie for your heart. A western tie are those like rope ties. So again, the thyroid gland is like a bow tie for your larynx, and it's spelled Larynx, L-A-R-Y-N-X. And then your thymus is like a western tie that sits on your heart. So let's go in general over the function of the endocrine system. The first thing that it does is it's going to maintain homeostasis. You know that homeostasis then is that metabolic balance that we see. It's how we keep specific levels of hormones or temperature or some kind of physiological system in check. Always remember, homeostasis is not a point, it's always a range. I forgot to take the noises off on this first one. The next ones won't have noises when they switch. The endocrine system is going to secrete chemical messengers. This is where you start filling in stuff if you haven't figured it out yet. Those chemical messengers are going to be called hormones. Hormones are spelled H-O-R-M-O-N-E-S. When we talk about the endocrine system, it's unique. Any system we talk about, it's good to list why they are unique because that tells you why they're different from other systems. The endocrine system is unique because it is ductless. Ductless is spelled D-U-C-T-L-E-S-S. Ductless means they are going to dump those hormones directly into interstitial fluid, then blood. So when we think about homeostasis, what does it do? Well, functionally, it's going to regulate metabolism. Metabolism is spelled M-E-T-A-B-O-L-I-S-M. So when we talk about the endocrine system, it's going to maintain homeostasis, that we know our physiological states. It's a range, not a set point. The endocrine system is going to secrete chemical messengers that are called hormones and that are unique because they're going to be dumped into interstitial fluid, then blood. And the main purpose in maintaining that homeostasis is to regulate metabolism. So now let's talk about endocrine reflexes. Remember, a reflex is an autonomic response. It's automatic. There are endocrine reflexes that are either simple or complex. It's easy to remember. A simple endocrine reflex has one hormone. You have organ 1 secretes one hormone that acts on endocrine organ 2 that has the correct receptor. When we talk about complex endocrine reflexes, they involve more than one hormone. So you'd have endocrine organ 1 is going to secrete hormone 1 that acts on endocrine organ 2 that's going to secrete hormone 2 that's going to act on endocrine organ 3. Might be easier if I gave you an example. The classic example would be your hypothalamus. Your hypothalamus is going to secrete regulatory hormones. So the hypothalamus is going to secrete FSH. FSH releasing factor is going to act on the pituitary gland to cause FSH to be released. So FSH releasing factor from the hypothalamus is hormone 1. It acts on the pituitary gland. The pituitary gland is going to secrete FSH. Besides simple and complex reflexes, we can look at how are these hormones built. So the first kind are what we call free hormones. Free hormones are going to be protein-based. They're going to be either amino acids, that's AA, or they're going to be peptides. They call them free because proteins are dissolvable in fluids, so that means you can dump them directly into the blood. When we talk about free structural hormones or the protein hormones, anytime you see F of X, it's like math. It means function. They function very rapidly, and they function very rapidly because they can get through the blood fairly quickly. Now, the second type of structural hormones are what we call bound hormones. Bound hormones are lipid-based hormones or sterols. Oil and water doesn't mix, neither does fat and blood. So when we talk about those bound lipid hormones, they have to be bound to a protein to make them soluble in blood because blood is mostly water. So when we talk about those bound hormones that are going to be lipids, they're going to last a lot longer than the protein hormones. And one of the unique things about the bound hormones is that they can serve as a reserve. You can store them. Okay, so we talk about endocrine reflexes, simple versus complex. Simple involves one hormone, complex involves multiple hormones. Structurally, they're either going to be proteins, they call those free because they can just go through the body systems by themselves. They act very rapidly because they can be released and start their work almost immediately. Bound hormones are those lipid hormones. You have to attach them to a protein so that they are dissolvable in fluids, and they are going to last a lot longer, and you can actually store lipid hormones where you can't store protein hormones. But the big reason we need to know the difference between lipid-derived hormones and non-lipid-derived hormones. Non-lipid obviously means proteins. These figures are going to be 18-3 on page 617. Let's look at lipid derivatives first. There's a picture at the bottom of the page there. When we talk about lipid-derived hormones or lipid derivatives, what's their importance? The importance is how they get in the cell. When we talk about their entrance into the cell, the lipid-derived hormones are normally going to be steroids. Steroids have to be bound to proteins when they travel through the blood. And why? We just said. They have to be bound to proteins because lipids are not soluble in water, which is what blood mostly is. So the protein that is bound to a lipid or a steroid hormone is what allows it to travel through the blood. If you look at the picture on the bottom there, F of X again means function. Lipid-derived hormones produce a nuclear hormone receptor complex. Okay, so let's look at that first. Now, we know that all cellular membranes are phospholipid bilayer, so that the lipid hormone doesn't need a receptor on the cell. It can go sliding right through. And then what it has to do is it's going to go through nuclear pores, and then it is going to attach to a receptor in the nucleus. So it's going to produce a nuclear hormone receptor complex. Well, once it binds to that receptor, it is going to activate genes for transcription. Remember, transcription is writing a message. Then you're going to have translation, and then you're going to have a response. So when we talk about lipid derivatives in the blood, they're going to be bound to a protein because that's going to allow them to transport. But to get into a cell, they slide through the membrane because they're lipid. They're going to go through the nuclear membrane, and they are going to make a hormone receptor complex in that nucleus that's going to cause activation of genes that's going to result in transcription, which results in translation, which involves a protein being formed. When we talk about that protein being formed, it's going to normally be an enzyme that's going to alter cellular activity because that's what hormones do. So when we think of the responses for, whacked my iPad. The response for lipid-derived hormones is always going to be 1-to-1. What does that mean? 1-to-1 means that you have one hormone, you're going to have one response because it can only generate one effect of one enzyme. So lipid-derived hormones, steroids that are bound to proteins in the blood, they make nuclear hormone receptor complex that are going to activate genes to produce a protein. That means that one hormone causes one response. That's very different from non-lipid hormones. We talk about non-lipid hormones. That means they're going to be proteins. Proteins can't slide through a membrane. So because of that, the hormone has to bind to a receptor that is on the membrane. That's very important. Protein hormones have to bind to a receptor that's on the cell membrane. That's their target cell. And that's just because they can't go through a cell membrane. The second thing that's going to happen is that it's going to activate a G protein. When we talk about activation of a G protein, that's going to be called a second messenger. In essence, one of the most important second messengers that we're going to find in the body is called cyclic AMP. It's abbreviated lowercase c, uppercase A, uppercase M, uppercase P. So when you activate the G protein, you're going to increase cyclic AMP levels. And cyclic AMP is a second messenger. When we talk about that second messenger, what is its importance? The example I always use is anybody that's ever had a job or everybody that has friends know you have that one person at your work or you have that one friend of yours that if you want a bunch of people to know, you tell that person. And if you tell that person, they're going to tell 9,000 other people. That's a second messenger. So when we talk about the importance of non-lipid derived hormones or protein hormones, they have second messenger effects. The importance of second messenger effect is that it's going to amplify the response. We talk about amplifying the response. That means that you're going to have one hormone and multiple responses. So when we talk about protein hormones, F of X, again, remember, means function. They're going to affect enzymes. Notice it says plural. It's going to affect multiple enzymes. Those will be activated. If you activate multiple enzymes, you're going to have multiple responses. They can affect cofactors, and those cofactors are going to affect ion channels. Just real quickly, remember that you can have coenzymes and cofactors that allow a better fit of an enzyme. When we talk about coenzymes, those are always organic. When we talk about cofactors, cofactors are always going to be inorganic. You think of something like calcium. So we have certain ions that can make that enzyme fit better. So let's compare the two. Both of them, the importance is how they get into the cell. Lipid derivatives, they're steroids. In the blood, they have to have a protein so that they're soluble in blood. Since they are lipid, they just slide through the membrane. They slide through the nuclear membrane. Then they're going to produce a nuclear hormone receptor complex. That complex is going to activate a gene for transcription. So you're going to make one protein. When we talk about lipid derivatives, their response is always 1-to-1. When we talk about non-lipids, they can't go through. Proteins can't go through a membrane. So protein or non-lipid hormones are going to bind a receptor on the membrane. It's going to activate a second messenger. Usually that's going to end up being cyclic AMP, and that's going to amplify the response. You're going to have one hormone and multiple responses. The function of those non-lipid hormones then is going to be to affect either enzymes or cofactors. Cofactors are going to be those factors that help ions work. And that concludes the first lecture.